Hello everyone, my name is Afeefa Zainab and I am master's student at Tohoku University, Sendai, Japan. Today the title of my talk is Characterization of Disease-Specific Genes and Molecular Pathways in Alzheimer's Disease and Huntington Disease. Before going into the details about the study, I would like to discuss a little bit about the diseases. So Alzheimer's disease and Huntington disease are complex brain diseases which share some of the common factors that include neurodegenerative. That is, the Alzheimer and Huntington disease both are neurodegenerative in nature, and also both of the diseases cause dementia along with depression. So these are the common factors or the common characteristics associated with both of the diseases that are actually one of the reasons of studying both of the diseases together, as there can be some of the pathways or genes that are actually associated in causing these phenotypes. So motivation, as I mentioned before, that because of the neurodegeneration of the diseases in the advanced stages, there is a very limited data available because of the postmortem changes. So most of the analysis cannot be done at advanced stages, and there is a great need to study these diseases in order to identify the complex pathways. And also another motivation behind the study was that the previous researchers mostly focused on separate studies of each of the disease. And therefore, we have focused on identifying the shared pathways and genes for potential drug targets in Alzheimer and Huntington disease. So differential expression analysis was performed for both of the diseases by comparing the sample, data sample of Alzheimer's disease with the healthy individuals and also Huntington disease with healthy individuals. This plot was generated using R through the Luma package. Here you can see that the blue region is indicating non-significant differentially expressed genes, while the green region is indicating the significant differentially expressed genes that are having a more or higher value of log to fold chain, while the black region is indicating the non-significant ones. So when we got the results of the enrichment analysis that we have performed using David, we have seen that the most of the pathways that were enriched by the upregulated genes of both of the diseases are similar. Here you can see that the pathways are showing high similarity with each other for the genes, upregulated genes, and similar behavior is shown for the downregulated genes as well. Here you can see that the pathways are almost similar, especially the top four pathways can be shown to seen to show a similarity in both of the diseases. When we were studying further, we found that the neurotrophin signaling pathway was the strongly enriched pathway in all the other pathways tested along with that. So we focused on the neurotrophin signaling pathway as it was having a very strong enrichment, and also when we have studied it further, it was having a very important role in both of the diseases as it is associated with the memory. And when we went into the detail for studying after getting the results from enrichment analysis, we have come to a conclusion that within the pathway, the BDNF, that is brain-derived neurotrophic factor, is one of the most important gene for regulation of this pathway. It was already studied that this gene is important for neuronal survival and also involved in learning and memory, and it has a role in Huntington disease. But we have found in this study that this gene is also downregulated in both of the diseases. So when we were identifying this disease, we have checked that whether this gene can be used for the therapeutic target or not. And we have found some of the studies where it is mentioned that this BDNF gene, this is the picture for neurotrophin signaling pathway. And here you can see that the targets, drug targets are labeled with the red, and the green one is indicating the available drugs. So it has been reported already that the immature BDNF actually initiates cell death pathway, that is neuronal death or neurodegeneration. So if we provide the adequate amount of BDNF, it can ensure neuronal growth, and also it can help to build up the memory in order to overcome the dementia or memory loss. So in order to identify further that if there is any specific gene available expressed in for Alzheimer disease and Huntington disease, or we can identify some unique genes, we have performed the protein-protein interaction network. We have actually extracted the protein-protein interaction network using string database, which was further interpreted, and these hub genes were extracted by using cyto-HUBA plugin from the Cytoscape. Here, these are the top 10 genes that are obtained from the Alzheimer disease, and these are the top 10 genes obtained from the Huntington disease. But still, these genes also have shown a bit of the similarity, indicating that the overall expression was very similar in both of the diseases in the prefrontal cortex region of the brain, which was studied. So to summarize the study, Huntington disease and Alzheimer disease shows a similar change of gene expression in prefrontal cortex region of the brain as we have found in the study. And another major finding was that the regulation of BDNF may play a significant role in pathology of both of the diseases, and also the pathways enriched by identified hub genes, if they would be further studied and they would be identified further, they can give a better insights in understanding the diseases at the molecular level. So that's all about my presentation today. Thank you so much for listening.